Alright, hey everyone, welcome back to the lectures, and today we're going to be talking about Chapter 8 from the book. So Chapter 8 covers basically metabolism. It covers the different types of energy, and how these things relate to the cell in general, and the whole thing that has to do with how energy is utilized. Okay, also like in the environment, not necessarily just a cell. All right, so let's get started. So the energy that's usually used by living things basically demonstrates the first law of thermodynamics. So thermodynamics, there are two laws that we're going to talk about today, uh, and they are the laws that basically are concerning how energy is utilized, okay, and how it is conserved, okay. So when we say that energy can be transferred or transformed only, but not created or destroyed, we're talking about the first law of thermodynamics. Okay, so it's kind of the same thing like when I told you in previous chapter when we were talking about like the chemistry stuff. Whatever reactants you have in the beginning, they are gonna be conserved, and you're gonna get basically kind of the same molecules and atoms, not molecules but atoms specifically on the product side. Okay, this basically the first law of thermodynamics is talking specifically about energy, though. Okay, so let's see here. So the conversion of energy to thermal energy released as heat by living things demonstrate the second law of thermodynamics. Okay, so that's the second law. Remember. So when for example plants, right, convert um, you know like thermal energy, or when it converts light, right, into chemical energy, there's always heat being released, okay, as a result. Same thing with animals, okay. Whenever we're trying to um, basically use energy, use like food for energy, right? And that energy that we do, right? Whether it's an action or simple walking or running or whatever you're doing, that releases heat, okay? And that scenario, that's specifically concerning the second law of thermodynamics, okay? And if I ask you on the exam, um, if I ask you specifically about the disorder of things or entropy, okay, you're gonna always answer that's concerning the second law of thermodynamics. Okay, so every energy transfer or transformation increases the entropy or disorder of the universe. Okay, so whenever you have energy that's being transferred, okay, so when we're talking about energy, we're talking about those atoms, right, and how they have their electrons, around them spinning, and all of these things. So these when that those these forces that are spinning basically are creating, energy, right? And whenever we have an increase of energy, we're increasing entropy. And entropy just means disorder. Okay, that's just the meaning of it in biology. Okay, so remember, if I ask you, energy that's being transferred or transformed, I'm talking about, how energy is conserved and it's not, created or destroyed. You're gonna answer me first law of thermodynamics. If I ask you how as energy increases, disorder or entropy also increases, you're gonna answer the second law of thermodynamics. Okay. Okay, so we're gonna just take a look at this briefly, okay? This is basically describing here on the left side the first law of thermodynamics, okay, that we just talked about. And then the one on the right side, it's describing basically the second law of thermodynamics, okay? So in regards to the first law of thermodynamics, and as I mentioned a little earlier, so the plants use the light from the sun, right? That light energy is being transformed by plants to chemical energy, right? Which specifically is being transformed to what? Do you remember? During photosynthesis, as being transformed to glucose or sugar, okay? And then when termites basically consume the plants, okay? So remember, the plants get the energy from the sun, and then the termites, which are basically other types of organisms, they're consuming those plants, okay, as part of the cycle of life, okay? So those termites are gonna basically utilize, right, uh, and make their own organic molecules, okay? Okay, and then basically you have the beetle larvae. This beetle larvae is consuming the termites, and it's transforming that energy, right, into a bioluminescence or light energy in the form of light energy. Okay, as you can see in this depiction. So you can see that this whole energy, how it started from the light all the way here, it's conserved. Okay, nothing was created along the way or someone. It was just transformed and conserved from one organism to another. Now, concerning the second law of thermodynamics, okay, we talked about how when an organism is using whatever fuel and creating energy, right? There's heat always that's being made. You see that? That's the plant using light to make organic molecules during photosynthesis. Heat is being produced as a result. Okay? Termite digesting plant and making new molecules, same way. Okay? And then that beetle larvae is whenever it's digesting those termites that it ate, it's also producing heat. And what does that mean? It's increasing the disorder or entropy. Okay. Okay, so we're gonna talk a little bit about metabolism. So metabolism specifically, is referring to the totality of an organism's chemical reactions. Okay, so that means the intakes and the outtakes. So whatever is being consumed and using whatever has been consumed, the organism transforms that into energy, or it transforms it also into simpler molecules, which it can use to rebuild uh, more complex molecules, right? All of these processes are part of the metabolism, okay? Metabolism is an emergent property. Remember that term from that we learned in the first chapter? So it's a specialty, right, that emerged uh, you know throughout the many years that have organism have basically evolved and all of that. So so basically it's an emergent property of life that arises from orderly interactions between molecules. That, as I mentioned, those interactions could be uh, during the intake process of digestion, or during basically the outtake, or whatever you're producing waste products out of that. Okay. Okay. So again, the metabolic pathway. So that process that we just talked about, where you have like a starting point, right? Okay. As you can see here, you have a starting molecule. Okay. There's several chain reactions sometimes that go along. Okay. um, and each step along the way is being catalyzed by a specific enzyme. Later on in this lecture, we will talk about what do enzymes actually do exactly, and how do they do their job. Okay, so enzymes are extremely important for chemical reactions. Okay, just to give you a brief idea, they speed up chemical reactions, and they're necessary for those reactions to move forward, okay, and finish fast, okay? So now we're going to talk about what kind of metabolic pathways do we have, okay? We have something called catabolic pathways, and we have anabolic pathways, okay? So the catabolic pathways are the pathways that basically release energy when you're breaking down complex molecules, okay? So what do you what kind of pathway is it when you digest proteins? Catabolic. What kind of energy is it when the body breaks down fat or muscle or whatever the case is? Catabolic. Okay. So um, cellular respiration is obviously a great example. The breakdown of glucose in the presence of oxygen. Okay. So what's happening during those cellular respiration, right? The cell is respiring, okay? What's happening is oxygen, right, is being used along with the glucose, okay? So the breakdown of glucose specifically. So breakdown of glucose from what? From whatever you consume, right? That, along with the oxygen, is a great example of that catabolism, okay? Now, the other pathway we talked about was anabolic pathway. Okay, anabolic pathway is the building of complex molecules from small ones, or simpler ones. Okay, so when we say we're synthesizing protein, a complex protein, okay, such as a muscle, for example, from the different small amino acids, we are talking about what? Anabolism or anabolic pathways. Okay, so it's the opposite of catabolic pathways. So just think about it however you want. So anabolic is the building. Catabolic is, I guess, breaking down of molecules. Okay. So this is just more stuff on it. Catabolic pathways are the downhill reactions, right? So you're going from a complex to a simpler molecules. The anabolic ones are uphill, okay? Um, and what do we know if we're going from low to an up? We need more energy, right, to make, correct? If you're trying to build a protein, a complex protein, right, in the case of anabolic pathway, you would want energy more to expedite more energy to be able to build that protein, okay? So the way it works is that organisms actually utilize both pathways at the same time, right? When an organism consume, right, certain food, it's basically using catabolic pathways to break down that food, right? And from that energy that's being released due to the breakdown of food, okay, and the simpler molecules that's being formed, the body uses that energy and those molecules to build and go uphill on those anabolic pathways and build again the complex molecules that it needs. Okay. So bioenergetics is actually the study of how that energy, okay, from between the catabolic and anabolic pathways is flowing throughout the living organism. Okay. So some vocabulary word, uh, you can basically tell what that means from just reading it. Okay, bioenergetics. So it's talking about the energy, okay, in biology or in bioorganisms. Okay. Okay, so let's talk about just define the energy. The energy is actually just the capacity to cause change. That's the definition of it. Okay, it can be used to do work. Okay. We can define it and move matter against opposing forces, like what? Gravity and friction. Okay? That's why when we talk about going uphill, right, you're using what more energy? Is that right? You're going against the the gravity. Okay, even in when we're talking about anabolic and catabolic reactions. So basically, energy, as you know by now, exists in various forms, right? Uh, just depends on what organism it is, what molecule it is, and all of that. Okay. So living cells, as you know by now, must transform that energy from one form to another. So in the case, for example, of us humans, right, we need to use glucose as the primary form of energy to produce the ATP and the different cycles that are known to be in the cell, such as like the citric acid cycles. So that's another thing that we're not going to talk about citric acid cycle, but that's just to give you an idea. Okay, so cells do use other certain types of molecules to transform it into whatever they need. Okay. All right, so let's talk about the different types of these energies. So the first one is kinetic energy. Okay, and kinetic, I don't know if you heard that term before. It's associated with motion. Okay, so whenever you have something moving, okay, you're gonna answer it's kinetic energy. Okay, so moving objects perform work by impacting motion to other matter. Okay, so an example would be when water gushes through a dam, basically turning the turbines. Okay, so the water force basically, okay, that's basically trying to put a lot of force on those turbines. They eventually turns on. Okay, so that's a kinetic energy. Now, as far as thermal energy, it comes from what? Thermal, so it has to do with what? Something with temperature, right? So is the kinetic energy associated with random movement of atoms or molecules? So what that means is when those atoms and molecules are moving due to energy, okay, what are they creating exactly? They're creating thermal energy. And in what type do they express it? Heat, right? Okay. So thermal energy is transferred from one object to another. It's called heat. Okay. Light energy is another type of energy that can be harnessed to do work such as photosynthesis. We know this by now, right? Who uses that light to that type of energy? Plants, right? They use it in photosynthesis to produce the different type of energy, which is what? Chemical energy, is that right? So they produce glucose, okay? Now, we also have something called potential energy, right? It's the energy that matter possesses because of its location or structure. So potential energy is usually something that's immobile, okay? It's not moving. So, for example, we have here water behind the dam possesses energy because of its altitude above sea level. So just the standing water behind that dam, okay, due to the differences in altitude, that's a potential energy, okay? Molecules possess energy, potential energy, right, due to the arrangement of electrons and bonds between their atoms also, okay? Now, when we talk about chemical energy, is potential energy available for release in a chemical reaction? As it sounds, right? What is a good example? We just mentioned it, right? Glucose being released during photosynthesis, that's a chemical energy, okay? Also, when glucose basically, such a, being a complex molecule, let's say in the body, is being basically broken down, right, to simpler products, then energy is released as a result too. Okay. And as you know by now, energy can be converted from one form to another. Okay. So, for example, chemical energy from food is used to perform the work of climbing up a diving platform. And we already talked about that, right? So you consume food, so basically to energize your muscles and the whole body to be able to do work, okay? Or kinetic energy, right? You see that? It's also mentioned here that the kinetic energy of muscle movement is transformed into potential energy. Oh, this is actually the example from the book. We have it here in this PowerPoint. Okay, just read that example where it's showing you the diver. Oh, here it is right there. Okay, so um, so this person, right, that's just standing on this platform here, has more potential energy on the platform than in the water, right? He's not moving, so he's got more potential energy. Okay. Now, diving basically converts that potential energy because it's now there's movement, right? It converts that potential energy to kinetic energy. See that? And you can see the climbing up stair converts kinetic energy of muscle movement to potential energy. Okay? Let's see if I should watch this.
Kinetic energy is the energy associated with motion. Energy that is not kinetic is called potential energy and is energy that matter possesses because of its location or structure. Energy is neither created nor destroyed, but converted from one form to another. This property is called the conservation of energy. Now let us see an example of how energy can be conserved. The bow and arrow start out with low potential energy. As the archer's arm pulls back the string, kinetic energy from the arm's motion is converted to potential energy in the tense bowstring and arrow. When the bowstring is released, the stored potential energy is converted into the kinetic energy of the moving arrow.
So the same thing here is happening with the person that was diving. Okay. So that arrow, once it was released, it converted that potential energy, which was basically when the person was holding the bow immobile. Okay, so it converts it into an actual moving arrow. So that would be what kinetic energy. Okay, and you can notice that when the arrow is actually flying, it produces what heat. Did you see that? Did you catch that around it? Okay. I don't know if you can show that again. Anyways.
It hits the target. Its motion ceases. If energy is neither created nor destroyed, where did the energy of the flying arrow go? Let us rewind and look at kinetic energy in more detail. When energy is converted from potential energy to kinetic energy, some of the energy can be used to do work. Some energy ends up as heat, a type of kinetic energy. Heat is the random motion of atoms and molecules. Watch the archer fire the arrow again. The red glow indicates heat. As the arrow flies through the air, heat is generated by friction between the arrow and air molecules. When the arrow strikes the target, all the energy becomes heat energy. The heat energy is rapidly transferred into the air and spreads out. Heat energy is a very disordered kind of energy. It has the highest amount of entropy or disorder of any kind of energy. Where do our muscles get energy to perform work, such as pulling back a bowstring? Our bodies use the chemical energy from food to perform work. Chemical energy is a form of potential energy. When your body breaks down food molecules, the stored potential energy from food can be converted to kinetic energy. Look at the archer's arm to see how energy is converted in a muscle. The stored chemical energy in food is released in your muscle cells during the process of cellular respiration. Using oxygen, cellular respiration converts chemical energy from food to another form of chemical energy called ATP, water, H2O, and carbon dioxide, CO2, are byproducts of cellular respiration. The potential energy of ATP can be converted to kinetic energy. Some of the kinetic energy is used to do useful work.
Okay, so hopefully this gives you a good idea of like a summary of all those types of energy that we were talking about. Okay. Alright, so let's talk about thermodynamics. It is the study of energy transformations. Okay. So in an isolated system, okay, that's just an example. When you put liquid in the thermos, okay, there is the liquid isn't able to interact with the surrounding environment, so energy is not exchanged. Okay. However, in an open system, right, we can see that the liquid will be able to interact with the surrounding, and thus energy can be actually transferred between the system, okay, and its surrounding. The system would be in this case, the liquid in the thermos, okay. So organisms are obviously in open systems, right? Otherwise, nobody will probably be alive, okay. They absorb energy from light or food. Um, we're talking about here who? We're talking about plants specifically, right? We as humans, we don't absorb really energy from light because we don't do photosynthesis. Okay, we don't have chlorophyll organelles to do photosynthesis. Okay, so we eat the plant, so that's part of that energy being transferred. Okay, um, and so the heat and metabolic waste such as CO2 um, are basically released to the surroundings as a result of that energy being absorbed. Okay. We talked about this already. So according to the first law of thermodynamics, the energy is what? Conserved. It's constant. Okay. It cannot be created or destroyed. Okay. You could call it the conservation of energy, and we talked about this already. Okay. You can see this bear is eating basically fish. Okay, and it's converting that nutrients in the fish to energy for it to be able to swim and do whatever it needs to do. Okay, so that's the first law of thermodynamics. Now in the second law of thermodynamics, you can notice that whenever this bear tries to run, right, it's creating what? Kinetic energy. Okay, and as a result of that, okay, what is the bear doing as well? He's respiring, right? So he's releasing carbon dioxide, right? Same as us. So he's breathing that oxygen and using the nutrients from the fish and converting that energy that he just consumed into CO2 and water, and as well as heat. Did you see that? Okay. And so what is this bear doing exactly? With this increased amount of kinetic energy, there is an increased amount of entropy through that heat. Okay? And so entropy is a measure of molecular disorder or randomness. Okay? Hopefully this is making sense a little bit by now. I know it's new stuff, but you just gotta study it and focus on it. You'll do well, for sure. Okay, so living organisms increase the disorder of their surroundings through their metabolism. Okay, so what does that mean? When we break down food, Okay, we're releasing energy and we're making small molecules such as CO2. Okay, that's just part of breaking down, the food. So that's metabolism itself. Okay, so processes that increase the entropy of the universe can occur spontaneously. Okay, so when we say spontaneous processes, okay, they're, as it sounds, they're spontaneous, so they occur without energy input. They're spontaneous. Okay, they can either happen slowly or quickly. Okay, so just think about this. Whenever you have um, increase in spontaneous, you have an increase in entropy. Remember this relationship. So processes that actually decrease entropy are non-spontaneous. Do you see that comparison? So if you have low entropy, it's non-spontaneous. You have high entropy, it is spontaneous. Okay? Okay, so just wanted to mention this, that if a system is spontaneous, okay, there's no energy input that's required. However, if it's non-spontaneous, okay, so that means it's not, done spontaneously. Okay, that means it's requiring an outside energy to be able to do um, whatever it needs to do. Okay. All right. So just wanted to tell you, and we mentioned this already, that how organisms create ordered structures. Okay, from less organized forms of energy and matter. Okay, so that we're talking about, for example, amino acids. We're talking about simple carbs like glucose. We're talking about nucleic acids. How all of these simple things the organisms use to build complex, um, you know, molecules like polypeptides, or even different body structures and molecules that it needs. It could be organs even, right? It could be muscle, it could be skin, whatever we're doing, right? So this is the example of building something very organized and very structured from an unorganized or less organized forms of energy. Okay. So the increase in order, what are we doing when we increase order? Are we increasing entropy or decreasing entropy? We're decreasing entropy. So entropy is the amount of disorder, right? Okay, so the increase in order within living systems is balanced by catabolic breakdown of organized forms of matter, releasing heat and small molecules. Okay, so you need those catabolic, we talked about this already, you need the catabolic breakdown of organized forms of matter to be able to build from these small molecules, you know, complex things, the things that you need uh, through the anabolic pathways, okay? So at a larger scale, basically energy is flows into the ecosystems as light and um, uh, exits as heat, okay? So the energy is flowing into the ecosystem as light initially, through photosynthesis, and it is exiting as heat. Okay, whenever those organisms perform kinetic energy. Okay. So just wanted to just read this, that biologists follow obviously the energy and entropy changes. Okay, both the energy and entropy or disorder quantity, right? Changes during chemical reactions to determine whether they require an input of energy or occur spontaneously. So what does this mean? We talked about how if it's if a reaction is spontaneous, what does that mean? It's increasing entropy, right? If it's non-spontaneous, it's decreasing entropy. Okay. So let's talk about this a little bit too here. So we're gonna talk about free energy. What does it mean? And it's denoted here as delta G. Okay. The G stands for the Gibbs free energy, and the delta means the difference between input versus output. Okay. So Gibbs free energy can be simplified and referred to as free energy. That's when we talk about Gibbs energy, that's why it's denoted by a G. Now, what is free energy, right, in itself? Free energy is basically the portion of a system's energy that can do work, okay? So it's a portion of a system's energy that can do work when temperature and pressure are uniform throughout the system as in a living cell, okay? So it's the ability to do work when the temperature and the pressure are basically uniform in the system. Okay. So change in free energy during a reaction is related to temperature and changes in enthalpy and entropy. Now I know all of these are new words, okay, but don't worry, we're gonna go through it little by little. So we talked about entropy already, what that means. Entropy means the disorder of something, okay. Enthalpy, okay, means the actual energy in a system. Okay, so that's just the total energy in a system. Okay, so there's an actual equation for that. So delta G or what free energy, right, denoted by the Gibbs free energy here, is actually equal to the delta H, or the change in enthalpy, which is the total energy, minus T is the temperature, right? Remember how we said that it's affected by temperature? Okay? Temperature, which is denoted in Kelvin. So Kelvin is being used in biology, which is basically a second form of temperature measurement besides Fahrenheit and Celsius. The way you measure it is you would add basically 32 plus 273 degrees, and you get the Kelvin, um, temperature that you want. Okay. And delta S, we talked about what is delta S, is the change in entropy. There we go. So entropy is S. The T is temperature in Kelvin. Delta H is enthalpy, or total energy in the system. And then delta G is free energy. So there's a difference here, okay? So the delta G for a process can be used to determine whether it is a spontaneous or not. And as you will find out that if it's a negative, so the result of this equation, whatever you're calculating, okay, if the result is a negative here, okay, negative delta G, it means that it's a spontaneous process, okay? And what does that mean? We have a higher entropy, okay? Now, if delta G is 0 or positive, that means it's non-spontaneous process, okay? So remember, we're going back to the slides before, right? And we said that non-spontaneous decrease entropy. Okay? Do you remember that? So I'm just trying to show you the relationships here between them. Okay? So every spontaneous process decreases the system free energy. Okay? So let's put it a little bit all together, right? If if we're talking about spontaneous process, we have a decrease in system's free energy. So we're talking about negative delta G. Okay, spontaneous is equal to negative delta G. Is also equal to more entropy, increased entropy. Okay. Now, when we talk about non-spontaneous process, we have a positive delta G, or 0 in this case, okay? And we have a decrease in entropy, okay? Let's see here. So delta G represents the difference between free energy of the final state and the free energy of the initial state. So that's also another equation for delta G. You have a final state, okay? That's why it's delta. Remember how I told you it's delta? So it's the G of the final state minus G of the initial state, okay? Because, in any type of, where energy is being expedited, right, or converted, or being used, right, you start with an amount of energy, and you either increase or decrease that energy, okay, depending on what you're doing in that system. So if a reaction has a negative delta G, right, the system loses free energy and becomes more stable. Okay, so let's repeat kind of everything together. So negative delta G is equal to more stability, okay, is equal to being spontaneous, yes, and is equal to increased entropy. Okay, now the other way is true. If you have a positive delta G, okay, the system is less stable, okay, you have non-spontaneous, normally, a non-spontaneous system, and you have less entropy, okay? So free energy can be thought of as a measure of system stability. We talked about this just a moment ago, right? You see that? So if you have a negative G, delta G, you have a more stable system. You see that? A lower G is more stable. A higher G is unstable system. Okay? So, for example, a diver on a platform, remember that picture we saw? When the person is still standing out there, is less stable than when floating in the water. That's just how it is, okay? Why? Because when he's in the water, right, the person is actually using kinetic energy to stay afloat in the water, okay? A glucose molecule is less stable than the simpler molecules in which it can be split. Okay? Just remember that equilibrium, as we mentioned this before, right? When we're talking about the reactants in the products, it's the point, same thing, at which forward and reverse reactions occur at the same rate. Okay, it is a state of maximum stability. Okay? Okay, so these are just, some examples when we're talking about the relationship of free energy to stability or capacity and spontaneous change. So this is kind of summarizing what we summarized just a slide ago. Okay, so in this situation here, right, when you have a person standing on that platform, or you have, certain molecule like glucose, for example, it's still intact. Okay, it hasn't split up. So you have these things are what you have more free energy here, which is a higher G in this case. Okay, it's less stable, Okay, and we talked about this here, right? That person is less stable here, and you have greater work capacity in this case. It's because, you know, the energy hasn't been expedited yet. Okay, now in a spontaneous change, right, what are we doing in spontaneous change? The system becomes more stable. The free energy becomes positive or negative? Negative, right? Do you remember that? The released free energy can be harnessed to do work, right? Same thing on the lower side here. Okay, when we split that glucose into the different small molecules, or when the gravitational motion is impacting the person when he jumps. Okay, we have less free energy in this case, which is a lower G. Okay, you have more stability in this case, right? And you have less work capacity as well. Okay, so this is kind of the same thing when we talk about chemistry of life processes, right? Same exact thing as I mentioned, right? So let's talk about exergonic and endergonic reactions in metabolism. Okay, so just right off the bat, exergonic means that heat is being released, energy is being released. Okay, endergonic, energy is being used. Okay, so definition is exergonic reaction proceeds with a net release of free energy to the surroundings. And this is an example of that top graph. Okay, so you start out with a whole more amount of free energy in those reactants, depending on what kind of reactants we're talking about. Okay, then as the reaction progresses, you have energy that's being released. Okay, when we see that, we're gonna say that this is an exergonic reaction. Okay, now whenever we're doing this, right, and we're releasing that energy, the difference between those reactants and products is basically the amount of energy that's being released, and it's normally a negative G, negative delta G. So it's basically a less amount of free energy because, you know, we started high, now we have a low products. Okay, so we have less amount of free energy. Okay. Now endergonic reactions, these basically are non-spontaneous. Okay, so you can just follow along, you know, and you can kind of tell how all of these are kind of related, right? So we said that negative delta G is spontaneous. It has more entropy, right? And non-spontaneous, we have a positive delta G. Do you see that? We're starting with lower amounts of free energy in those reactants, and basically we need energy. For example, if we're building like you know, amino acids into a complex protein, right, as a product. So you need energy, right, to be able to build that. So that would be what a positive delta G, or it would be non-spontaneous because you're requiring energy. Do you remember that? So we said the non-spontaneous, you require energy. The spontaneous do not require energy. Okay? You guys can watch all of these. They're on the PowerPoint. Okay. So in exergonic, this is just an explanation of what I just explained. An exergonic reaction, the products store less free energy than the reactants. Okay, we can see that from that graph. Because delta G is negative, okay, we already talked about this. Exergonic reactions occur what? Spontaneously. Okay, do you see that relationship? Okay. Now, endergonic reactions, right? The products, sorry about that. The products actually have more energy, more free energy specifically than the reactants. Okay, so this would be, for example, a complex protein molecule, and these would be, for example, amino acids. Okay, so you need energy to build those uh, complex molecules that have a lot of more free energy than reactants in this situation. So this would be a what? A non-spontaneous and also a positive delta G. Okay, so let's talk about just this as an example, right? So reactions in a closed system eventually reach equilibrium and can then do no work. So what this is showing you here is that there's a chamber filled with water here, okay, and as the water rushes in spontaneously through the turbine that you see here, it it basically allows that turbine to do work, and you can see how the light is on in this situation. Okay, it keeps doing that until basically you see here what you see here. When both chambers are equal, that work will eventually stop, and that's what we call equilibrium. So in this situation here, right, the flow of water, basically spontaneously, that would be a delta G, that's a negative. Okay, do you see that? It's less than zero. And whenever we have both chambers, that's equal or zero or neutral, that's when there's no more work that can be done. Okay, so let's see here. The chemical reaction of metabolism are reversible, as we know, right? You can go from complex molecules to simple molecules, um, but just so you know, in living organisms, cells will never reach equilibrium, and for a purpose, right? Because if they do, they will die. If the intakes versus the outtakes are equal to zero, then basically the cell will die. Really, it will have no job to do anymore, no work, and thus it will die. Okay. Um, so comparing that to electric system, as you can see here, we talked about this upper one already. So that's an open hydroelectric system that we just talked about. And then this is a multi-step open hydroelectric system. This is kind of what's going on in the cell. Okay. So one spontaneous reaction drives another, and that drives another, and so on. And the cells will never reach this phase, which is equilibrium. Okay, you will always have that negative delta G. Okay. So a cell does three main kinds of work. Okay, just so we know this. So it does chemical work, okay, which basically doing endergonic reactions. So it's um, you know, building molecules and so on. It basically transport work, okay. So it pumps substances against the direction of spontaneous movement. Okay, do you remember those gates, protein gate channels that we talked about? They require ATP, and so these pumps, they're going against the direction of spontaneous movement, or um, the concentration gradient. Okay, it does mechanical work. Okay, so that means doing contraction of muscle cell, or of whatever, like I guess nerve cells. Okay, nerve cells move along the axons. Okay, so to do work, cells manage energy resources by energy coupling. So as it sounds, it's energy coupling. So it's using a couple of things to accomplish um, the work. Okay, so it's using an exotonic process to drive an endergonic one. Same thing as when we talked about catabolism and anabolism, same thing. Okay, so we use exergonic process, which releases heat and energy to drive, using that heat and energy to drive an endergonic one and build whatever molecules we want. Okay, and as we know by now, ATP is the energy molecule of cells that does that. You guys can watch that on your own. So ATP, adenosine triphosphate, okay, is this cell's energy shuttle, as we said. Okay, it's made out of what? You remember? Adenosine, that would be an amino acid. Okay. I mean a base, sorry about that. It's a base, nucleic base, and then you have a ribose, a sugar, right? And you have the three phosphate groups that are attached to each other. Okay, so the way cells use ATP is when they hydrolyze the ATP molecule you see here as an example using water. Do you remember in hydrolysis we use water to break down things? And so whenever hydrolysis happens, we end up with ADP, or adenosine diphosphate. So you have two phosphates instead of three. And you have a single inorganic phosphate as a result. And that breaking down of that phosphate away, it releases what we want, which is what? Energy. Okay? Okay, you guys can see that. So we talked about how the breaking down of this terminal phosphate, okay, using hydrolysis, it releases energy. Okay, so the energy does not come from the actual phosphate bond itself, but actually from the chemical change to a state of lower free energy in the products. Okay, so what this means is we're going from a bigger molecule which has more free energy to a smaller one, and as a result we're creating energy. Does that make sense? We're releasing that amount that was stored in this and separately. That's why we're breaking down the phosphate. Okay. So a repulsion between the negative charges of the three phosphate groups. Okay, so they have negative charges here. Let me show you that. You see that on the O's? On the oxygens, okay? The repulsion between those negative charges of three phosphate groups create a lot of potential energy, okay? We already know this, right? ATP is powering all of those types of work for the cell, mechanical, transport, and chemical, okay? In the cell, the energy from the exergonic reaction of ATP hydrolysis can be used to drive an endergonic reaction. We already talked about this, okay? It's just repetition for you to understand. Just so you know, the overall coupled reactions are always exergonic in cells. Okay, just like it's kind of always negative delta G, same way. Okay. So whenever the cells are using ATP and they're splitting that phosphate atom or element away from that ATP molecule. They use the process phosphorylation. It comes from phosphate. Do you see that? Phosphorylation is the transfer of phosphate group, right, from that ATP. So it's that phosphate group. We're transferring it to another molecule, okay, to be able to power an endergonic reaction. That's what the cell wants to do. That's why it needs energy. Okay. Now the recipient molecule, we call this a phosphorylated intermediate. So it has already been phosphorylated with that phosphate. So we call it a phosphorylated intermediate is more reactive, okay, because it gained energy, right? So it's less stable, of course, and it's got more free energy, okay? And there's a lot of things going on here, but let's see here. So looking at just the A, it's just showing you an example of how the cells use ATP to do an endomorphic reaction, or you're building basically a complex molecule from simple molecules. So we're talking about here glutamic acid. Okay, that's a simpler molecule, and ammonia. We're using them to build glutamine. Okay, and to do that, okay, so endergonic reactions give you what? A positive delta G. Do you remember that? While exergonic give you what? A negative delta G. Okay, so looking at B, right? They're just showing you how glutamic acid uses ATP. Okay, do you see that? It gets phosphorylated using when that phosphate basically gets attached here, and we call this phosphorylated intermediate now. And due to this ATP losing this phosphate, it becomes ADP. Do you see that? And so now we add the ammonia, and we have glutamine. This is what we're looking for, right? So looking at basically the free energy change for the coupled reactions here, okay? So the way we're going to look at this is we have basically the endergonic reaction and then the exergonic reaction. So what we did here is we used these two simpler molecules, the glutamic acid and ammonia, okay, to build this glutamine. To do this, we coupled it, right? This is the coupling reaction with the hydrolysis of ATP. Do you see that? ATP was hydrolyzed to ADP and a phosphate. When this happened, so we broke down that ATP into two simpler molecules. What did we do? We released energy that was used to build this molecule. Okay, hopefully this is making sense a little bit. So when we did that, we broke down those into simpler molecules. We actually did what? An exergonic reaction. Okay, what do we know about that? It's a negative delta G, negative free energy. Okay, now if you were asked what's the net of these, total, like what was the energy exponential, you're gonna basically subtract. Okay, it would be basically the plus 3.4 that you got from the endergonic, okay, plus the minus 7.3. So that would give you a total of negative 3.9 kilocalorie per mole. Okay, so the overall of this would be, do you remember what we said? The overall of a coupling of endergonic and exergonic are what? An exergonic reaction. Okay? So you see how you have a negative delta G? Alright, so so how the hydrolysis of ATP performs work, okay? The transport and mechanical work in the cell are, as you know by now, they need ATP to be hydrolyzed to be able to do that. You need the energy from ATP, okay? I'm sorry about that. So ATP hydrolysis, whenever it's being broken down, leads to a change in the protein shape and binding ability. So let's look at this protein basically carrier that's actually embedded. It's an integrin in that cell membrane. Do you remember that? So um, we're looking at a transport work and we're looking at a mechanical work examples. So for transport, you can see that for this protein to carry this molecule, it needs to be phosphorylated. Do you see that phosphate here? So it's using ATP to be able to change its shape and then transport that solute or molecule over to the other side. Also, to do mechanical work, um, the same thing is going on here as well. So a motor protein in this case, like if we're talking about muscle cells or nerve cells or so on, okay, or cytoskeletal tract in this case, you need that ATP as well to be phosphorylated here to be able to also let that vesicle move on that cytoskeletal tract. Okay. So ATP is a renewable resource, right? That is regenerated by addition of a phosphate group to ADP, right? So without the phosphate, ATP becomes ADP D as in David. If you wanna make it into an ATP again, you're gonna add that phosphate again. Okay. Free energy needed to phosphorylate ADP comes from exergonic um, breakdown reactions catabolism. Okay, so you can see how this system is actually a closed system, right? They're working together to basically help each other, both catabolism and anabolism, um, to do what needs to be done. Okay, shuttling of inorganic phosphate energy is called the ATP cycle. Okay, so it couples energy yielding processes to energy consuming ones. Okay, we already know this by now. So in order for us to build something, we need to consume something, and the reverse happens again. So energy from catabolism, basically, okay, when we're basically breaking down molecules, we're releasing energy. That's called an exergonic energy releasing process, right? Okay, when that happens, we're gaining ATP, right? And for example, when you eat bread, right, you're creating ATP. Okay, then ATP with water through hydrolysis, okay, uh, is being used when a cell goes and does basically an endergonic reaction, okay, um, which is the energy consuming process. It uses that ATP through the phosphorylation and to do whatever it needs to do. So it's a process, it's a cycle that goes on and on. Okay, so we're going to talk about enzymes a little bit now. So enzymes, I said this before, they speed up metabolic reactions. We do need them. They're very necessary, required, by lowering the energy barriers. Okay, they cannot do anything else to the reaction. They do not add or subtract. Okay, they can just speed up a reaction. Okay. So for example, the hydrolysis of sucrose to glucose and fructose is spontaneous. Um, at room temperature, a solution of sucrose in sterile water would sit for years without appreciable hydrolysis. Okay. However, if you add sucrase enzyme, okay, the sucrase is going to be able to basically break it down into the glucose and fructose simpler molecules. Okay, oh, here we go. So this is showing you exactly what we just said. Okay, so an enzyme is basically a catalyst. A catalyst is a chemical agent that speeds up a chemical reaction, right? We just said that, without being consumed by the reaction. Okay, so it just speeds it up. Okay, and this is what we just talked about, how sucrose is, being aided by the enzyme sucrase to be able to be broken down into glucose and fructose. Okay, so in order for the enzyme to do what it needs to do, it needs to lower something called activation energy. So let's go through this little by little. So every chemical reaction between molecules involve bond breaking and bond forming. We already know this by now, right? A molecule must be contorted into a highly unstable state before bonds can break can start the reaction. And you're seeing this here a little bit. So you start out with reactants here. So A attached to B, C attached to D. Okay, and you have this much there here as far as free energy. Now, in order to break down these bonds, okay, we want to basically have a certain amount of activation energy to be able to start, um, getting those bonds unstable to be broken down. You see that those products, the bonds are different than these. They're not the same. You see that? Okay. So here is where enzymes come. The enzymes, what they do is, you see that amount of, it says here EA, which is activation energy. Okay. What they do is, whenever you add an enzyme, they lower that graph like that. So because of that, less amount of activation energy is needed, and as a result, okay, so you don't need to reach that threshold to be able to activate and break down those bonds. So as a result, the reaction speeds up. Okay. Let's see here. An exergonic reaction, right? The formation of new bonds releases more energy that was invested in breaking the old bonds. Do you see that? You have less free energy here. Okay. So therefore, there's more, energy being released out. Okay. You see how the delta G is below zero, so it's a negative delta G here. So adding heat is not a useful way to speed reactions. Heat usually what it does is it really speeds up atoms around, and as a result, speeds up the energy. But cells can't use that because as a result, proteins will be denatured because of the heat. Okay, so here's where enzymes come in. So enzymes do not require the addition of heat. They themselves lower the activation energy, as I mentioned. Okay, and as a result, when that activation energy is lowered, the reaction proceeds much faster than before. This process is called catalysis. Okay, the process by which a catalyst, such as an enzyme, selectively speeds up a reaction without itself being consumed. Okay, remember, an enzyme cannot modify the reaction. It cannot change the free energy from like a positive to a negative delta G. You can see that on your own. Okay. So those enzymes are not just like random, they're very specific for certain substrates or whatever they're trying to react with or to speed up the reaction. Okay. So the reactant that an enzyme acts on is called the enzyme's substrate. That's the name of it. Whenever they're attached together, after the enzyme reacts with it and binds with it, we call this enzyme substrate complex. So they're complex. They're formed by the enzyme plus the substrate. While bound, the activity of the enzyme converts substrate to product. Okay, so this is what we want, right? We want to convert and speed up the reaction. using the enzyme. This is just showing you exactly what we just talked about. So here's a substrate. Here is the big enzyme. Okay. This substrate is supposed to fit on that right there. So that's usually for every enzyme has a specific site for that fits that substrate, kind of like a puzzle. Anytime you see a word ending with ase, it means it's an enzyme. Okay, like sucrase, for example. It's an enzyme to break down sucrose. Okay. Let's see here. So again, it's a complementary fit, right? When the substrate enters the active site, okay, and it's bound here, it changes the shape of that enzyme site slightly, okay. Basically, it tightens around the substrate like a handshake. Okay, so holds it very tightly. This induced fit, that's what we call this induced fit, results from interactions between chemical groups on both the substrate and the active site. So you can imagine, there are chemical bonds happening here that basically holding on that substrate in its place. Okay. You can see that on your own. Okay, so the substrate is typically held in the enzyme's active site by weak bonds, okay, such as hydrogen bonds. Okay, so whenever the substrates are here, they're held by hydrogen bonds. Okay, do you see that? What did we say happens? Whenever the complex happens, okay, we do what we need to do by lowering the activation energy. Okay, the substrate, or being what a product now, not a reactant anymore, is released, and so uh, the active site is now available for new substrate instead. Okay. All right. Let's see here. Okay, so the active site on an enzyme can lower the activation energy barrier by orienting substrate correctly. So this is ways how the activation energy is actually lowered. It's shown you why this happens. By straining substrate bonds, okay, by providing a favorable microenvironment, right? So all of these are happening in that binding site, and by covalently bonding to the substrate, okay? The rate of an enzyme catalyzed reaction can be sped up by increasing the substrate concentration, okay? So if the enzyme is saturated, right, the reaction rate can only be sped up by adding more enzyme. So that means, if you have a lot of substrates, okay, you have certain amounts of enzymes, sometimes, you get a limit or a maximum amount where of these substrates are occupying these active sites. So you need more enzymes to have more additional active sites to be able to speed up the reaction more. Okay, it just depends on how many substrates, the balance between the enzymes and the substrates in the system in the reaction. So an enzyme activity can be affected by environmental stuff like temperature and pH. Okay, you have higher temperature, it might increase the enzyme's activity, it might decrease it. You have an acidic or a basic environment, it's going to do that as well. Also, there are some chemicals that specifically influence the enzyme. Okay, each enzyme has an optimal temperature, right? So there's a specific temperature that this enzyme likes to work at. It's very specific. Okay, up to this point, the reaction rate increases with increasing temperature. Beyond this point, the rate of reaction begins to drop. So just looking at this graph right here, you have on the x-axis you have temperature in Celsius, and you have on the y-axis the rate of reaction. And so the blue graph here, line graph, is actually showing you the optimal temperature for a human enzyme, which is under 37 degrees Celsius. Okay, and now the optimal temperature of enzyme of thermophilic, heat-loving bacteria, it's working at a higher temperature. Do you see that? So it just depends on what environment this enzyme is made for. Okay, and basically the rate of reaction increases with increasing temperature, and we can see that, right, until it gets to a point where the reaction starts dropping. Okay, you can read all the details on your own if you want to study everything. And again, you have also optimal, pH environment, right? Same as the temperature. So an enzyme might like a lower pH, like pepsin in the stomach. Another enzyme might like, a higher pH, a basic one, like trypsin, okay, which is an enzyme. So it just depends on, the environment. Now, cofactors are, like aids to enzymes, right? So they're non-protein helpers that bind to the enzyme permanently or reversibly with the substrate. So they help enzymes. Inorganic cofactors include metal atoms, okay? So usually those cofactors are made out of zinc, iron, copper. So usually all those vitamins and things we consume, such as these, are basically helping enzymes. Okay, they're cofactors. Organic cofactors are called coenzymes. So if they're organic, we're going to call them coenzymes. Okay, so there are actually chemicals that actively inhibit enzymes from doing what it needs to do. So if an inhibitor basically forms covalent bonds with the enzyme, then the inhibition is usually irreversible. Covalent is normally like stronger bond, okay? So many inhibitors bind to the enzyme by weak interactions instead. And they do that to be able to be reversed. Okay? Whenever the cell wants, or whatever body part wants that to be reversed, they can get reversed. That's why they don't do the covalent bonds. Okay, hang in there. So we're almost there for that PowerPoint. So let's talk about the competitive inhibitors, right? These, as it sounds, they compete. Okay, they compete with the substrate. Whenever you have those competitive inhibitor, they basically come and lodge in that same site, okay, and prevent the actual substrate from binding to the enzyme. Okay, as a result, the enzyme productivity is reduced. Okay, increasing the substrate concentration can overcome this type of inhibition. So if you increase this substrate, okay, I mean there's like more of that competitive inhibitor, then there's more chance for those substrates to actually bind to the active sites of enzymes. Okay. Now, non-competitive inhibitors. Okay, so they're not competing. There's no competition between the non-competitive inhibitors and the regular substrates. So these, instead, what they do is they bind on a different site beside the the actual site they are trying to block, okay, where the substrate binds. So such as in this case. So that inhibitor binds on the other side of this enzyme, which basically signals this enzyme to change its active site shape, and because of that, the substrate does not fit um, like a puzzle anymore. So it blocks basically this enzyme from doing what it needs to do. Some examples of these inhibitors would be toxins, poisons, pesticides, and antibiotics. Sounds familiar, right? You can watch this on your own. Um, Toxins and poisons are often irreversible enzyme inhibitors. For example, that's in the book. Sarin gas was used in a chemical attack in Syria in 2017, killing many people. Sarin, what it did is it binds covalently to the active site of acetylcholinesterase. Okay, so this is an enzyme that's being used in the acetylcholine site between where the nerve ending and the muscle um, connects. Okay, so that serine will block that enzyme, and so you have a problem basically in the nervous system, um, getting its signal to other parts of the body. Okay. So let's see. The regulation of enzyme activity helps control metabolism, right? Chemical chaos would result if a cell's metabolism pathways were operating simultaneously. Sorry. Okay. So they have to basically operate one at a time. Okay. Cells can regulate metabolic pathways by switching on and off genes that encode specific enzymes, right? So maybe we'll want to speed up this reaction now. Maybe we want to shut down this reaction. So they code for specific enzymes to regulate, all the activities that are going. All right, so let's talk about the allosteric regulation of enzymes. So allosteric regulation may either inhibit or stimulate an enzyme's activity. Okay, these regulation occurs when a regulatory molecule binds to a protein at one site and affects the protein's function at another site. Kind of similar to the non-competitive inhibitor. Okay. So looking at this here, so this is an enzyme, right? It's got different active sites here. Okay. So whenever we have an activator here that basically binds to it, the whole enzyme becomes active, as you can see. Okay, in the active form. Now, if we have an inhibitor, right, that it binds to just one specific place, all of the active sites become inactive, basically. Okay. So the binding of an activator stabilizes the active form of the enzyme, as you can see here. The binding of an inhibitor stabilizes the inactive form of the enzyme. Okay. In cooperativity, okay, substrate binding to one active site triggers a shape change. Okay, kind of what we saw earlier. So it just binds to one site. Okay, which triggers a shape change in all of the sites that stabilizes the active form for all other sites. Do you see that? So it was inactive without this substrate, and now it's active. Okay. One more thing about how enzymes are inhibited. It's called feedback inhibition. So you have an initial substrate, let's say, it binds to an enzyme, right? And you have more production of like intermediate productions of those enzymes. And when you have like a lot of these, you know, and you have an actual like the end product, which is the product that you're looking for, then this end product sometimes will do a negative feedback, which basically this isoleucine in this case will bind to that um, enzyme, and then it will basically inhibit it. Okay, so it will have a place for it to bind. That's where what we call allosteric site. Okay, so it binds to the enzyme on this side, but it blocks, um, from further blocks like more threonine from binding to the active site. Okay, so finally we're going to talk about how the cell basically has compartments in it that helps bring order to metabolic pathway. Not everything is like, all over the place, okay? Everything is organized in the cell. Some enzymes act as structural components of membranes, okay? In eukaryotic cells, some enzymes reside in specific organelles. And you're looking at a mitochondria here. You can see that the matrix contains enzymes, right, that are involved in the second stage of cellular respiration. As you know by now, the ATP, the energy form of that the cell uses, is produced mainly in the mitochondria. This is where cellular respiration happens. So because of that, enzymes are needed for those reactions. Enzymes also can be found in, embedded in this um, inner membrane inside also the mitochondria for the third stage of cellular respiration. Okay, and that's it for this chapter. I know this was a lot of information, a lot of new stuff, but just study it, guys. Go over it, and let me know if you have any question. And I wish you all the best of luck. Have a good one. Bye bye now.